Diethylsulfate polyacrylamide gel electrophoresis (Sds-Page) is a cornerstone technique in biochemistry, molecular biology, biotechnology instrumental in the separation and analysis of protein based on their molecular weight. This method is essential for various applications, including protein characterization, purification, and the study of protein-protein interaction. It plays a pivotal role in assessing the purity of protein samples, verifying recombinant protein expression, and identifying disease biomarker, and also profiling protein in different biological samples. Sds-Page is crucial for its precise resolution of protein mixture, enabling identification, quantification, and analyzing of protein isoform and modification. In protein purification and characterization, Sds-Page is crucial for assessing the purity of protein sample and determining their molecular weight, essential for understanding protein structure and function. In molecular cloning and expression studies, it verifies the expression of recombinant protein, ensuring they are correctly produced in sufficient quantities. In clinical diagnostic, Sds-Page is identify disease biomarker and analyze protein pattern in samples for diagnosis and disease monitoring. In proteomics, it profiles protein across sample, aiding in the study of expression, interaction, and cellular process, advancing understanding of disease mechanism. The principle behind Sds-Page involves the denaturing of protein using SDS, an ionic detergent that impacts a uniform negative charge to the protein, thereby masking their negative charge, linearizing them. When subjected to the electric field in the polyacrylamide gel matrix, this protein migrates to the positively charged anode. The rate of migration is inversely proportional to their molecular weight, with smaller protein moving faster towards the gel pores and larger protein migrate more slowly. The size-based separation allows for the detailed analysis of protein sample. The Sds-Page process comprises several key steps. Initially, proteins are denatured by mixing with SDS and a reducing agent like beta-mercaptoethanol or DTT to break the sulfide bond in sample preparation. In gel casting, polyacrylamide gel are then cast with varying concentration to create a suitable matrix for the desired resolution. During electrophoresis, samples are loaded into the gel walls and electric current is applied, causing the protein to migrate. Post-electrophoresis gel are stained with dyes such as cohesive brilliant blue or silver stain to visualize the protein bands. Finally, the gel are destained to remove the excess stain and the bands are imaged and analyzed. Sds-Page is versatile and can be applied to various samples, including cell lysate from bacteria, yeast, plant, or animal cell, purified protein for molecular weight determination, serum and plasma for diagnostic purpose, tissue extract to study tissue-specific protein expression, and recombinant protein from different expression systems.
For sample preparation, a loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue, and glycerol is added to the protein sample. Protein samples may be biologically derived, for example, from prokaryotic or eukaryotic cell tissues versus environmental samples of purified proteins. When the sample buffer has been added, the samples are then heated at 65 degrees Celsius for 5 minutes.
Gel casting process regular preparation, a separating gel solution with pH 8.8 and stacking gel solution with pH 6.8 are used. Both consist of alkaline ammonium presulfate and TUKIMIN. The gel will produce by polymerization between two glass plates anchored vertically in a cassette. First, the separating gel will be poured. The polymerization reaction is a vinyl addition polymerization initiated by a free radical generating system. Ammonium presulfate is used as free radical indicator, while TUKIMIN accelerates the rate of formation free radical from presulfate. This free radical will convert alkaline monomers to free radical, which react with inactive monomers to begin the polymerization change reaction. The gel, characterized by its characteristic porosity, is formed by randomly cross-linked elongating polymer change by bis alkalamide. The gel's pore size is influenced by the ratio of alkalamide to bis alkalamide. Low concentration is preferred for high molecular weight sample. The stacking gel solution is poured on top of the solid separating gel. During polymerization, a plastic comb is placed on the top of the stacking gel to create small wheels within the gel. After the gel polymerized, the comb is removed and the gel cassette is vertically placed between two electrodes. The positive electrode is located at the bottom of the gel, while the negative electrode is located at the top. Gel is placed in a chamber and a pH 8.3 lysine chloride buffer system is poured to facilitate current condition through the gel. SDS is present in the gel and running buffer to ensure that the protein remain denatured throughout the run.
For sample loading, in addition of the samples, a molecular weight size marker is loaded onto the gel. Next, using a micropipette, carefully load each protein samples into the wells of the gel. As we have seen previously, bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is useful for visualizing the samples in the well, and glycerol increases the density of the samples and used to make the sample fall to the bottom of the sample well rather than just flow out and mix with the running buffer.
Now we move on to electrophoresis. After loading up the sample into the well, apply electric current to the gel. Make sure that the wire is inserted correctly. After turning on the power source, protein will migrate from the negative electrodes towards the positive electrodes through the separating gel. Smaller proteins will move through the gel faster than bigger proteins. Therefore, the bigger proteins will always be behind the smaller protein. Stacking gel is very important to ensure that all the proteins in the well start at the same position before entering the separating gel. If the stacking gel is absent, all the proteins will be unsorted upon entering the separating gel, which will result in a smeared band. The bromophenol blue has a smaller molecular size than proteins, which makes it move faster than the proteins. Electrophoresis will be stopped when the migration front reaches the end. Afterwards, all proteins are sorted by size and will be analyzed by various methods such as Western blotting. After the glass plates are removed, the well and stacking gel will be removed. And then, a generous amount of Kumasi blue dye will be poured. Afterwards, the separating gel will be soaked into the Kumasi blue dye. After the proteins are stained, the proteins can be detected as blue bands on any clear background. The proteins in the sample can be compared with the protein ladder to know the approximate size of the protein in the sample as they travel the same distance, which means that they have the same molecular weight.
Western blotting is a widely used analytical technique in molecular biology and biochemistry. The process begins with sample preparation, where the cell or tissue are lies to extract proteins. This is often done using a buffer that contains detergent, salts, and protease inhibitors. The protein concentration is then measured to ensure that the equal amount of samples are loaded onto the gel. In the gel electrophoresis step, proteins are denatured using sodium dialyl sulfate SDS, which imparts a uniform negative charge to the proteins. These proteins are then separated by size through polyacrylamide gel electrophoresis page. The gel may be stained to visualize all proteins before the transfer. Following electrophoresis, proteins are transferred from the gel to a membrane made of nitrocellulose or polyvinylidene fluoride (PVDF) in a process called blotting, driven by an electric field. The membrane is then incubated with a blocking buffer to prevent non-specific binding of antibodies. Next, the membrane undergoes antibody incubation. A primary antibody specific to the target protein is applied, binding directly to the protein of interest. After washing away excess primary antibody, a secondary antibody is added, which binds to the primary antibody. This secondary antibody is usually conjugated to an enzyme like horseradish peroxidase, HRP, or alkaline phosphatase, AP, for detection purposes. Detection involves adding a substrate for the enzyme linked to the second antibody, resulting in a chemiluminescent or colorimetric signal. This signal is then captured using imaging equipment such as a chemiluminescent detector or X-ray film. The bands corresponding to the target protein can be analyzed and quantified to determine the relative amount of protein in the sample.
Similar to how Western blotting detects specific protein in a sample, a mass spectrometer can also identify protein. However, a mass spectrometer is more precise as it can quantify protein molecule by measuring their mass to charge ratio. A mass spectrometer operates through 5 stages: vaporization, ionization, acceleration, deflection, and detection. During vaporization, the sample is heated to convert it from a liquid into a gas. In the ionization chamber, the vapor is bombarded with electrons from the electron gun, resulting in the formation of positively charged ion by removing the electron from the molecule. the positively charged ion then accelerate towards the negatively charged plates. When these ions enter the mass analyzer, they are deflected by the magnetic field, with the degree of deflection depending on their mass to charge ratio. Consequently, they travel through the mass analyzer at varying speed. Finally, as ions of different mass to charge ratio reach the detector sequentially, a computer generates a spectrum of ions present in the sample, displaying their mass to charge ratio on the x-axis.